Aloha and good morning, Hawaii. It is December 30th, 2020. I'm Ryan Kalei-Suji, joined by Yunji Denise. Yunji, it is our last show of the year, and it's been quite a year, but we are continuing on in our discussion, of course, of the issues that are impacting Hawaii, and we continue to talk about COVID-19 and a potential new development that could help in this battle against the coronavirus.
That's right. So important to battling the coronavirus has been testing, contact tracing. And one thing that could make it so much easier would be to have rapid testing that perhaps you could even do at home. So joining us this morning is Dr. Pat Sullivan from Oceanit. He is the CEO and founder of that company based right here in Hawaii. They have been working on a saliva test that we have been watching very closely because Dr. Sullivan, this could really be a game changer. Thank you so much for being here this morning. For our audience who may not be familiar with Assure-19, please tell us a little bit about the technology, how it works, and where you are right now in production.
Well, thanks, and thanks for having me today for an update. Yeah, the technology, so this is an important part of, for I think folks here especially to understand. We started about 6 years ago creating an artificial intelligence that's more like a human, that deals with ambiguity, small data. It's called strong AI. We refer to it as anthronoetic artificial intelligence. And about 3 years ago, we started working with and collaborating with DARPA on using it for understanding genomics, trying to read a genome like a language, looking at the letters, kind of the way we look at an alphabet, to see if we can derive intention. And the long-term goal is to have a causal connection versus correlation in biology. So, can we cure cancer, that kind of thing. So we then, when this started, we pivoted the tool set we created, which included what we've referred to as the grammar of RNA, to design a molecule specifically for COVID-19, or a piece of it, that we could then use as a test. And it turns out there's a lot of the virus. You have it in your nose, you get it in your chest, but you have it in your mouth. You have a lot of these ACE2 receptors in your gums and your saliva glands. So we thought if we could develop a simple saliva test, and where you can get a result in about 10 minutes, then you'd have a real handy tool that you could use on a regular basis. And there's something endearing about sticking a thing up your nose that after a few times, you kind of get a special feeling, where I think compliance might be an issue, where spitting in the morning is not hard for a lot of people to do. So it's a spit in a cup test. But what we do is you spit, we collect the saliva, and then you run it through this process. You shake it a few times. It's got an electrolyte. All this work, by the way, is all invented and created here in Hawaii. So we began clinical tests with Queens, and we've been collaborating with University of Hawaii School of Tropical Medicine, and we got really good results. Then we kind of went into this next phase of producing more tests, because scale becomes a big issue. As you might have seen, there's all kinds of papers and studies out there that show that you need 10 to 30 million tests a day just in the United States. And this is going to go on for quite some time. So we thought, okay, we need to figure out how to go to scale. We've been trying to set it up here, but we started collaborating with, there's a bunch of companies that have reached out to us, and one's in Korea. And so they sent us some of their first run. They were really good. Then they sent us another batch, and they were terrible. And so we've been trying to sort out what happened. And that becomes a critical piece of getting the FDA approval, because the quality systems and reproducibility. And so we were kind of locked and loaded to just wrap the whole thing up about 3 weeks ago. And then we just kind of hit this wall. And, on one hand, it should be expected. You're learning new things. And the way to think about it, perhaps, is what we need is a big bakery. We don't have a big bakery in Hawaii, so we found one in Korea, and we're teaching them how to make our bread. And we make it here, and it's awesome. But when we went there, they did exactly, we think, the first time around. Then they started to scale, and we think they swapped out ingredients, maybe put in olive oil instead of margarine or something. And all of a sudden, it's different bread. And so we're working through that. The issue, though, is that all of this is created right here in Hawaii. All the technology is homegrown, and we think there's a real need, and you can use it in the visitor industry here. And gosh, there's so many ways to use it for opening schools and that kind of thing. But we need to be able to go to scale. And to do that, you're going to produce it in a factory of sorts. It's a big, clean room. There are molecules, there's things like that, but it's things people could be trained on and could work very well with, I think, our local community. But that's where we're at. We've been in discussions with FDA. We know what they're asking us to hit in terms of the benchmark. They've never done anything like this. All the tests they've approved are different. It's a single-stranded DNA molecule that we've invented. This is something out of science fiction for most folks, but was really all created here. So what they do now is they use antibody tests, and there's PCR tests, which you've heard about, which are very sensitive. But this is in a whole different category. So they're excited about it. There isn't a saliva test like this in the world, so they're excited about that. But getting the recipe right so we can produce the bread and scale it, that's kind of where we're at.
Let's talk a little bit about that FDA approval process. So where are you at now in terms of getting that approval? Let's say you were able to figure things out on the manufacturing end. About how long do you think that takes before the FDA is able to officially give you the green light to go ahead?
Well, the rule of thumb people are using it for this group. We're part of this National Institutes of Health RADx group. So it's a down-selected group from around the country with promising technology. And they're saying we could probably do it in 30 days. Now, we were on a call with the director who actually makes the decision. Former professor from Duke was pretty intrigued by all the stuff we're doing. What we're trying to do is just stay focused on one piece of the test. This will be a CLIA wave test. What that means is that you could do it if you've got a medical personnel in charge. You could do it at the airport, you could do it at schools, you could do it with emergency response folks. But you need to have somebody with a little bit of medical background, essentially. The one we're trying to get to is an over-the-counter test. And they're excited about that too, but we need to produce a lot more, and we need to be able to produce them all with the right kind of quality. And so therein lies kind of this issue of going to scale, because an over-the-counter test needs to work for all kinds of people pretty much anywhere. And so it's got to be exactly the same every time.
Let's talk about that over-the-counter test. What would the price point for something like that be, and where do you see it being used? Is this something that you would take every morning, or something that you would only take if you were getting on an airplane? What do you see the actual application for something like this, and how much would it cost?
Good questions. It's not expensive to make these. So we've said the going out right now is like $20, but when you go to scale, you could be $5. I mean, it doesn't have to be expensive, but the question about how you would use it. So there's a bunch of really interesting studies, and what you've got going on is this transformation with the administration in DC. The current administration has not been very keen on testing, because the more you test, the more you find, and they didn't want to know about that. The new administration has put out a lot of material about testing in schools, testing and testing and testing. So this rapid testing. And there's data that shows if you take 10 to 20% of the population, test them daily, you can wipe out the virus in a community pretty quickly. And so what this will come down to, I think, is creating bubbles and bubbles and bubbles until you've got one big bubble of Hawaii, and the whole country could do the same thing. And so the analysis and the math around that, the economics around that, you've got all kinds of people that have been promoting that idea of rapid, easy, affordable, accessible testing. So to your question, you've got different scenarios. You get off an airplane. What we've been doing with our folks that got to travel is we call the Aloha protocol. You do 5 days of testing. And so you test every morning. And if you're negative, you can wear a mask, go about your business, no problem, because your risk of infecting anybody is quite low. After 5 days, it's pretty much zero. because the virus, after exposure, peaks at about 5 days. Symptoms show up maybe 2 days later. So we've kind of seen this reproducible pattern. The big problem, of course, is roughly half the population are asymptomatic spreaders, meaning they just feel fine, but they're spreading to everybody around them. And so that's the big problem. And you don't know if you're asymptomatic. You just think you have a kind of a morning funk and you need more coffee or something, when in fact, you might be fighting the virus and not know it, and you could unwittingly spread it to your family and friends. So the idea is to go through this process of testing, and if you're negative every day, because it goes up so steeply once you start getting sick, you're able to stay safe and keep people around you safe. Just a case of a little bit of detail. You infect other people roughly around 1 times 10 to the 6 viral particles a milliliter. Milliliter is a tiny amount of fluid. So that's a viral concentration. We would measure it way below that, but when you're at that number, you start infecting others. Maybe not too much, but you start to. And then, of course, in a couple of days, you're way up there. It goes up, like logarithmically. So it could be 10 to the 11th viral particles. That means you're a super spreader, right? So what you're doing is you're catching it before you even are able to make others around you sick. Now, getting sick is about your immune response, your level of health, your comorbidity status, all those kind of things. But there's these rough numbers, rules of thumb that you could apply that say, if I can catch it low, then I take you offline if you're positive and you don't infect anybody. But if you're showing negative, even if you've got it kind of coming, but it's just not enough to express itself, we'll track you the next day and the next day. So after 5 days, you cover pretty much the run of the virus going way up and pull them offline. That's how you would eliminate infectiousness in a community. And there's some really good studies, and there's a professor at Harvard, Mike Mina, who's kind of the evangelist out touting this from the mountaintops for anybody that'll listen. But I think the new administration is listening.
We want to bring in an audience question from Edward Jones here, who's asking, on a previous interview, you mentioned that there was DoD provided manufacturing lines. Still a possibility? I think it goes back to the questions about how we are able to, or how you folks are maybe able to produce this, not only in Hawaii, but here in the United States. Is the DoD still an option? And what are some other options beyond just going internationally, but maybe staying within the country?
Well, good question. We have a close relationship with DoD. They identified a facility in Maryland that we could basically ramp up. I think what they're doing is kind of waiting for us to get through the EUA before we do that, because the different combatant commands are interested in using this, because you could have a bunch, throw them in your locker, and if you do a shore deployment from, let's say, a ship, you come back, you can start testing. And so there's some really simple ways to use it. It's a pretty, since we're using these single-stranded DNA molecules, it's more resistant to just sitting around on a shelf. It's in an airtight packet. You just open it up, kind of like, I don't know if you've had these electrolyte drinks, right? If you're working out, you sweat a lot. They come in these packets, a lot of them, and you just pour them in water. But if you leave them out, they absorb moisture. This thing's in a little airtight packet. So you could throw them in a locker and they could stay there for a long time. So yes, there's that facility. Most of the infrastructure around the US, however, is way overcommitted. The capacity is very low. And so that's where we thought there was an opportunity to build something here, because a lot of these kinds of tests over the last, say, decade, used for pregnancy and veterinarian applications, that kind of thing, have been low margin medical technologies. And so a lot of companies, the big companies that used to do this, they sold those businesses, and a lot of that went overseas. So the actual capacity in the US is small because it's really moved out. We've got a big, really a company that was put together by a hedge fund, I think, but it rolled up all the European and American small versions of this, and they're interested in scaling up with us too. They have some facilities in the US, but Europe is their biggest market. So yeah, it's kind of an interesting problem because companies are going as fast as they can, but the infrastructure in the United States for this kind of thing is kind of thin.
I wanted to go back to something that you were just talking about creating these bubbles and the application of the test. So on a practical level, let's say we wanted to do a school and then do that community and so on. Do you then, when you talk about testing 10 to 20% of the people or of the population, do you then only test 10 to 20% of the students, or do you test everybody for that sustained 5-day period and then expand the bubble and on and on? How does that work?
Good question. That's how I would do it. I would test every kid, everybody in the school, and do it for 5 days to create a bubble at the school. And then the question is, when the kids go home, have they created a bubble at home? Right? So are the parents controlling the environment so that you don't have any, when Uncle Bob comes from Las Vegas, right? And Uncle Bob is sleeping in Johnny's bedroom. All of a sudden, you just introduce something and you got to track Uncle Bob, right? Because Uncle Bob is the new change in the system. And so the bubble at home is important, but I would do the bubble at school, establish the bubble at school. Once you do this for, say, 5 days, then the frequency of testing at school would go down. You could test every couple days, every 3 days. You're just trying to find any outliers, but it's important that the bubbles at home and the bubbles that support the school bubble all are part of managing this program.
In terms of the availability, of course, it was one thing, but when we talk about the masses, so let's say that there's a sporting event at UH and we want to welcome fans back into the arena. Is that something that you could foresee fans taking this test in order to get in, or to a concert, or to any sort of large gathering? These obviously would be people outside of their own bubbles, but it would present an opportunity for people to get tested to make sure that they are negative before entering in. Is that something that you foresee being a positive of this?
Absolutely. That's a future scenario. We've talked to people about the Olympics in Japan. That's exactly what they're interested in. They want to have the events. Everybody coming in gets tested. And if it's a simple saliva test, then it's relatively easy. It's interesting, we've been doing for a future version what's called an orthogonal test, which is doing a nasal version of our test. And one of the guys, we were talking about it because for little kids and for events where you've got a range of people, he said you include the nasal test and you mix it in with tears, because if you just keep sticking up your nose, you do it until you cry. And that's one of the challenges with the current methodology, even though it's not bad. Obviously, the deep nasal one is the standard that people have been using for PCR, and then there's a shallow one that people have been trying. But ultimately, you'd want to test people at big events. You could do it at restaurants. You could do it at concerts. And what that'll do is it doesn't make it zero risk, but it significantly reduces the risk. And it's really about risk management. So what you're doing is you're reducing risk. Everything has got risk. But if you reduce risk and say everybody in this bubble has been tested, so, be smart, but everybody should be in pretty good shape in this area at this time.
What's the accuracy of the test?
Well, a PCR test is more accurate. And the way they measure accuracy is with a level of detection. So level of detection, accuracy is referred to when you're doing the FDA thing is how it compares to a PCR test. And we're trying to be 95% as good as a PCR test within a certain range. And we've gotten really good results to show that that's the case. But a PCR test does a thing called amplification. So it's looking for a strand of RNA, and it cycles for hours and hours and hours looking for it. And that's why those tests can be more accurate. But you don't really express virus after exposure for maybe a couple days. So even if you're exposed, the PCR test won't show that you got it because you're not producing anything it can really measure. And then you've got this problem after you've expressed virus and you no longer are producing virus, you still might have remnant RNA strands in your system. Those will show up on a test. Those are called false positives. So each test has its own challenges, but the sensitivity of a PCR test is better than, and it's really the gold standard. So we're pretty darn good. I mentioned the viral concentration of 1 times 10 to the 6 is where you make people sick. We're probably 1 times 10 to the 4 in terms of viral content, which means a PCR test will pick up right around maybe 1 times 10 to the 3. It's kind of right around there as the threshold of what it can actually see. So we're not far off, and it takes you about a day or more to kind of start getting up that curve from there. So for our test, you get it before you're at a risk of making everybody sick. It's got enough sensitivity to do that as an operational tool.
You know, I want to ask, with the rollout of the vaccine that's currently happening, and you know, people are getting encouraged to take the vaccine, how does that impact your overall plan and rollout? Would that be something that you would still encourage people, although they had a vaccine, to still take this test? And would those who maybe have a vaccine not take it, and only those who have not are the ones that would be the ones that would take this test?
Excellent question. So there's a variety of things, you know, kind of to unpack around that, but there's some unknown things about this. So bottom line is, if you get vaccinated, be careful, be diligent, because first, you don't know how long it confers immunity, assuming you get 2 doses. We just haven't been tracking people long enough to know, you know, is it relatively short term or is it a few months? And you see people getting reinfected. There's just a lot we don't know because it's happening so quickly. And there's a concern that it will only confer immunity for so long where you might need a booster later, like some of these other, you know, diphtheria, polio, other things. So that's a possibility. Second is there's a concern that you could have a vaccine and still spread it like an asymptomatic spreader. And so you could make somebody else sick without even knowing it. And these are unknown, right? These are questions that we hope CDC and others will kind of bear down on. And then there's this question that you see in the news that there was just a news account where it showed up in Colorado, this mutant strain. So there's nothing unusual about a virus mutating, right? If you make enough copies of anything, at some point it starts to kind of get a little tweaked and changes. And through evolution, this is how things evolve to make it more efficient. So they say it's a little more infectious. Doesn't surprise me, because if you're a virus, you're trying to evolve, again, if you're kind of following more of our Darwinian evolution, to be more infectious. So that's not a surprise. The real question is it at risk of making you sick again? And how serious is it? So there's a bunch of questions there about this new strain that I think require further study. It's not that we're living in a science fiction twilight zone. It's that this is what viruses do. And so getting our heads around the science is important. And that's where an outfit like the CDC is so critical, because they should be bearing down and answering these questions, which then goes to the bigger question: would you still want to do this test if you get vaccinated? I think the answer is yes. You periodically check yourself to make sure that you're not asymptomatic, you haven't picked up something else. So, you know, you could pick up a slightly different strain and have some weakened immune response as one scenario, but you could still have it, you could still produce it. And based on your current medical condition, there could still be a risk. So we have to work through these issues.
I know we talked about the FDA at the top. Councilmember Carol Fukunaga is joining us this morning. She asks, how long do you think it will take for the FDA to act on your application for emergency use authorization? I guess the question everyone wants to know is, when can I buy this?
Thank you, Carol. And thanks for all the stuff you do, Carol. I mean, Carol's a real warrior for the community. I think we'll be pushing to get it in January. We kind of lost the bubble in December because we had this batch come from Korea that it's not unusual for this kind of thing to happen. It's just we're trying to go so fast. So we had a whole bunch of them just not work. We were kind of in shock because we thought, like, we're right at the finish line. We're done. Because we had done all this work with Queens, collected data, put all this stuff together, and we were going to do a kind of a final run with some hospital patients and others. And it was like, wow. It didn't get out of our labs because we couldn't get past the quality system, our own quality system, but that's what happened. And so we're working through it. We're trying to come up with possibly some makeshift ways to produce here in the interim, but we don't have, again, it's like a factory to make bread, right? If you're going to produce a lot of bread, you need a big enough factory to produce it. And we don't have that here. So we're trying to figure out how to do it with others, and we'll do that. The tragedy there again is the lack of, you could produce jobs for people that could, help diversify the economy. And there's a future for the things we created, this platform to design a molecule for a test, a therapy, everything from more future viruses to anti-cancer treatment, diabetes. There's all kinds of things you could use it for.
Sorry, I'm muted. One last time here in 2020 where my mic is muted and I'm talking. Dr. Pat Sullivan, we thank you so much for taking time to update us. And I know Yunji and I, and as well as many people out there, are looking forward to hearing some positive news and some updates that will essentially get this test in hand to people in Hawaii sooner rather than later. So we wish you all the best of luck. And thank you so much for updating us on where you guys are at right now.
Thank you.
Thank you. Aloha. And
now I'm muted.
We both ended.
Exciting news, and really unfortunate to hear that they had that hiccup that he was just talking about with the manufacturing in South Korea. But they are working really at warp speed. And January, I mean, that's just in a couple of days. So it does sound like they are very close to being able to get the approvals that they need and get this to market. And he was saying that, this could really have broad application for schools, as you noted, for sporting events. It just could really be a game changer. And production at anywhere from $5 to $20 per test would really be something.
Yeah, and just another sort of a weapon in the arsenal of battling coronavirus and all that's happening, of course, with the vaccine. And we heard his explanation about that, and also suggesting that even if you're vaccinated and have gotten those 2 shots, that this is something that you could continue to use. And we could see it being something that businesses or organizations require for people to take before entering a facility. And so it does have some promising side, and we have been looking forward to this for some time now, as we talked about this test over the past few months here on The Conversation. But we look forward to hopefully hearing some news here from them in the coming weeks.
Yeah, and we've got a great January scheduled for you folks already. We are looking ahead to the new year, putting 2020 behind us. Next week, of course, there's going to be a change this weekend when we see the new mayor being sworn in, and the Ledge is coming back into session very shortly. Speaker Saiki is going to be joining us on Wednesday to talk about the outlook for the legislature. And then Governor Ige is joining us on Friday. When we had talked earlier just this week, it sounded like they were going to be able to delay furloughs. We got news that it looks like furloughs are being delayed until at least July, thanks to the new CARES Act funding. So much to talk about with the budget and the outlook. And then later on in the month, we've got more exciting guests.
That's right. And it's hard to believe we've already come to the end of what has been a crazy year. And we've certainly enjoyed experiencing all of this with all of you, the viewers here on the platforms of the Honolulu Star-Advertiser. We want to take a look back now, a quick look back at some of the highlights from our time here with all of you on Spotlight Hawaii.
I'm Yunji Denise, joined by Ryan Kalei-Suji, and of course, this is Spotlight Hawaii. Governor, good morning.
Mayor Derek Kawakami.
Lieutenant Governor Josh Green. Good morning, Dr. Char.
Peter Ingram.
It's Mayor Kirk Caldwell. He is joining us live from his office.
Rent. But many of the bigger land.
You're getting tired of watching Tiger King, and
I don't know if anyone's getting tired of watching Tiger King. It's pretty interesting.
Getting shedding any kind of red abatement.
From Dot Chang. Happens to be my mother, Washington Hero.
I knew. I sincerely apologize. There's a princess in our midst.
In a large lecture hall, if you can remember that far back, Ryan.
We hope to see all of you here in Hawaii at some point as things sort of lighten up.
David, Governor. Question.
I forget what the governor has dubbed this new period. Care for others, live with aloha, something along those lines.
Hard to believe that's been our year here on Spotlight Hawaii, and it really has been a crazy year, and great to see that as sort of we've had a number of guests who have joined this conversation, and we thank all of you for being involved with it, and all of our guests who have been a part of this sometimes crazy moments here, even on our show.
Yeah, we've really enjoyed it. We started this back in March, and we thought, okay, let's just do this for the few weeks of lockdown. And now it's extended. The coronavirus has really taken our state on such a crazy journey. And we appreciate all of you being here. We appreciate all of your questions. And we are excited that the paper has committed to continue this conversation into the month of January. So thank you all of you for being a part of it. We have really enjoyed having you all with us along for this last year.
Yeah, and as we sign off here as our final episode of 2020, and looking forward to 2021, we wish you and your family all the best here as we head up into a new year. We hope that you will stay safe during this holiday season. Of course, a time where we normally will gather with family and friends on New Year's Eve, but hope that we can all really be cautious, listen to what officials are saying us, limit our group sizing, wear a mask, and continue to stay safe as we battle this and go through this time together. So until next year, I'm Ryan Kalei-Suji, joined by Yanji Denise, and we'll see you right back here on Monday here for Spotlight Hawaii. Aloha.
Happy New Year.